Well, the beginnings of a new day here, and it's actually going to be a pretty big day today because we're going to talk to a handful of faculty about the model systems that you use in their laboratories and in their research. So I've got you here with me in my bathroom this morning because we're going to try to give you an idea in terms of how accessible specific organisms are in your life, the things that are around you every single day. And with that, we'll take some samples. I'm thinking there's one more place we should stop that'd be relevant to who we're going to visit with first today, and that's Professor Dan Kearns, who studies Bacillus subtilis in his lab. So let's head into work. So on my way into work here, I've stopped right in front of Jordan Hall. We're actually going to take a soil sample right here, right in front of the building. Question is, what kind of bacteria is in here? We're in the lab of Professor Dan Kearns, and we're going to look at what we collected from our samples this morning, from my toothbrush, my mouth, and from the soil in front of Jordan Hall. So let's get started. Now what's really cool about this is we've plated out the bacteria from our soil sample as well as from my mouth and from the toothbrush. But something that needs to be considered is, can all bacteria survive the same environment or the same conditions? And what's really cool about Bacillus subtilis is what Dan studies, is that it's very resistant to heat. In particular, its spores are extraordinarily resistant to heat. So what I'm going to do right now is we're going to take these bacterial samples that I took from the soil, and we're going to heat them up. And we're going to compare what grows in that media before heat and after heat treatment. Okay, the 20-minute incubation is up, so let's take them off our heat block and plate them. Put them in the incubator. So I'm here now with Professor Dan Kearns from the Department of Biology, and he's going to tell us a little bit about his model system today and a little bit about his research. So Dan, my first question for you is, what is it about your model system that got you excited in terms of the research that you do, and what are the strengths of that model system?
Well, I think that the main strength of our model is just how well-developed genetic system that it has. It's probably the best understood bacterium after E. coli, so we have access to a wide range of genetic tools, a complete genome sequence, and it's just very easy to manipulate.
I see. So, and where do these bacteria come from, the ones that you study in particular?
This bacterium comes from the soil. You can isolate it pretty much any soil sample out in the environment. It's probably living out there, or it's probably existing out there as spores, but those spores are just very abundant in the soil.
And that's something that actually I just did myself, so we'll get to look at those results in a second. The next question is, in terms of overall strengths of your system compared to maybe other systems, I mean, is it expensive to grow? How accessible is it? Obviously we can find this in the soil, so it's one of those things where it's very easily accessible. And even if you think back to the early 1900s when, you know, the forefront of genetics and us not even really understanding molecular biology yet, what are the powers of it innately?
I think that the Bacillus subtilis system has two primary strengths. One is just simply the speed at which it grows. It doubles about every 20 minutes in the laboratory. What this means is we can start a culture in the morning and by mid-afternoon be working with it. And just that speed of growth makes it very, very simple to deal with. Or you can at least do a lot of experiments in a short amount of time. The second aspect is that it has a property, it's called naturally competent. This means that it can take up DNA free from the environment and incorporate it into its genome. This means we can just take the cells, add our DNA, and have those cells incorporate those constructs. It makes it very, very easy to genetically manipulate. In fact, it's so easy to manipulate that the focus of my lab is to no longer use these very easily amenable laboratory strains, and we're actually going back to study strains that are more closely related to those that are taken directly from the environment to see what types of behaviors those have.
So bacteria from literally the real world and not just the lab strains that we work with here.
Yeah, we'd like to think so.
That's pretty exciting. So in that regard, are there weaknesses? Are there things that you feel like you can't study with your system, and what are the things that you'd like to be able to do? What are the things that make it maybe slightly more difficult to work in your system than others?
I think that the Bacillus system is sufficiently advanced that really the only limitation is imposed by us. We have all the tools. All we have to do now is come up with new ways to use them and design new approaches. But really that limitation is on us, not on the system.
Okay, that's pretty cool. The final question I have is, a lot of laboratories use more than one model system, and that might not necessarily be for research, but for tools in general. Do you utilize any other model systems in your research?
Yeah, we use Escherichia coli, probably the workhorse bacterium out there for doing molecular genetics. We build our constructs there and move it into Bacillus. We've started using Saccharomyces cerevisiae, common yeast, to do things like two hybrid analysis to detect whether or not two proteins interact. And we're also now branching out into another yeast system called Pichia, and this is a robust protein expression system.
Very cool. Well, this has been Dan Kearns. He's told us a little bit about Bacillus subtilis, the model system he uses in a laboratory, and I want to thank you for your time today, Dan.
Thanks, Joe.
Appreciate it.